Okay, so welcome to this next video in which we are discussing the formation of a tertiary lymphoid organ in a chronically inflamed tissue. Okay, so we've seen how T cells that are within the inflamed area, okay, so these could be T helper 1 cells, T helper 2 cells, or cytotoxic T cells, they have lymphotoxin beta on their surface, which is going to activate the lymphotoxin beta receptor on the endothelial cells. Okay, we've seen how this causes type 2 activation of endothelial cells. Now what we want to look at is another pathway which is going to lead to the transformation of the endothelial cells in the postcapillary venules into high endothelial cells, which is going to make them have a very similar structure to the high endothelial venules that you have in lymph nodes, which serve to assist naive T cells and B cells in moving from the blood into the lymph node, and also in moving from the lymph node to the blood. So when this transformation happens in these capillaries, in these venules within the chronically inflamed tissue, it leads to naive T lymphocytes and B lymphocytes being moved from the blood into the chronically inflamed tissue, and therefore you get the development of this tertiary lymphoid organ, as it's called, which is this mass of naive T cells and naive B cells that is within the inflamed tissue. Okay, so let's now discuss the pathway that through which lymphotoxin beta causes this transformation. Okay, so we'll go over the page for this, because it's a big pathway. So, here is the lymphotoxin beta receptor, and it has now been activated, let's say, by the lymphotoxin beta on the surface of a T cell. Okay, so this is the lymphotoxin beta receptor, LT beta R for short. Okay, so what is going to happen is we're now going to discuss the non-canonical NF-kappa B pathway. So, this lymphotoxin beta receptor, when it's got lymphotoxin beta bound to it, which I'll just show there, okay, is going to activate a kinase enzyme known as the NF-kappa B, or nuclear factor kappa B, inducing kinase. Okay, so let's put this enzyme here. So, this is the nuclear factor kappa B, the NF-kappa B, inducing kinase. Okay, and for short, that's often abbreviated to NIK, N-I-K. So you take the N from NF-kappa B, the I from inducing, and the K from kinase, and get NIK. Okay, so this enzyme is going to be activated by the lymphotoxin beta receptor once it's got lymphotoxin beta bound to it. Now, what does this enzyme do next? Well, again, it's going to act on the same enzyme complex as we saw in the canonical NF-kappa B pathway. Okay, so it's going to act on this complex involving the inhibitor of NF-kappa B kinase enzymes. So remember, this enzyme complex consisted of the inhibitor of NF-kappa B kinase gamma, okay, IKK gamma. It also consisted of the inhibitor of NF-kappa B and then kinase alpha, IKK alpha, and also the inhibitor of NF-kappa B kinase beta. Okay, and previously we saw how the MEK3 enzyme and the TAC1 enzyme in the canonical NF-kappa B pathway phosphorylated the inhibitor of NF-kappa B kinase beta. Now, this NF-kappa B inducing kinase is going to phosphorylate the inhibitor of NF-kappa B kinase alpha rather than the inhibitor of NF-kappa B kinase beta. So we're going to stick a phosphate group onto the inhibitor of NF-kappa B kinase alpha. Now, that is going to activate this enzyme, but in a totally different way. The target of this enzyme is going to be totally different from before. Okay, so its target is going to be a different NF-kappa B enzyme. Okay, so let me show you this other NF-kappa B transcription factor. And I'm sorry I said enzyme, it's a transcription factor. Okay, so this other NF-kappa B transcription factor consists of a protein known as P105, okay, and also another protein, let me just bring this over, known as REL-B. So remember, the normal NF-kappa B, the main NF-kappa B transcription factor, consisted of P65 and P50, okay, and P65 was also known as REL-A, and P50 was also known as NF-kappa B protein 1. Okay, now we've got this other NF-kappa B transcription factor, and by the way, the NF-kappa B is the entire thing, so the whole thing would be called an NF-kappa B transcription factor, but it's not the main one, it's a sort of off-the-side one. Okay, right, so this is a nuclear factor kappa B, however. Okay, so what is this enzyme complex going to do to this NF-kappa B? Because it's in the cytoplasm at the moment, so it can't go into the nucleus, it's not active yet. So, what the enzyme is going to do is add a phosphate group to P105. Oh, and what you also have to understand is that this doesn't have the inhibitor of kappa B like the P50, P65 dimer did. Okay, so what is stopping this from being an active transcription factor? Well, it's the structure of this NF-kappa B transcription factor itself, so it needs activating, basically. So it's going to have a phosphate group put onto this P105 by this enzyme complex here. Okay, so when you put a phosphate group onto the inhibitor of NF-kappa B kinase alpha, it adds a phosphate group onto the P105 protein within this NF-kappa B complex. Okay, so what's going to happen now is that P105 is going to get proteolytically cleaved once it has that phosphate group on, and it's going to get proteolytically cleaved down to P51. So P105 changes, basically, it's cleaved down, and it now is P51, so you chop a bit off, and it now is P51 rather than P105. And it's still bound to the REL-B. Okay, so now this is the active transcription factor, basically. This was the precursor, this is the active transcription factor, and this conversion is a proteolytic cleavage. Okay, right. So, what happens next? Well, this is going to go into the nucleus and activate the transcription of certain genes. But just before we discuss that, let's just say that P51 also has another name. It's also known as NF-kappa B protein 2. Okay, so just as P50 was the NF-kappa B protein 1, P51 is the NF-kappa B protein 2. Okay, so this P51 REL-B complex is now going to go into the nucleus. It's going to bind to the promoter regions of certain genes and increase the expression level of those genes. And the expression of these genes is going to produce the changes in the endothelial cell that make the endothelial cells now resemble this high endothelial cell instead. So it's going to cause them to go from having a flat phenotype to having this tall phenotype. Okay, now one particular protein that I want to talk about because it's well characterized is a protein known as high endothelial cell associated N-acetylglucosamine 6-sulfotransferase. Okay, so this is a huge, great mouthful. So, one of the proteins which is going to have its expression enhanced by this transcription factor, and which is very, very important for tertiary lymphoid organogenesis, is this high endothelial cell, and don't worry, there is a acronym for it, high endothelial cell, and all of those are with dashes between them, associated N-acetylglucosamine 6-sulfotransferase. Okay, and I will explain exactly what this is going to do, because it's quite interesting how it works, because this enzyme is going to be responsible for making the changes to the polysaccharides on the surface of the cell that are going to allow these naive T cells and naive B cells to get across the endothelial cells, to stick to the endothelial cells, and then transmigrate across them or through the gaps between them and get into the interstitial space and then form a tertiary lymphoid organ. Okay, so N-acetylglucosamine 6-sulfotransferase, and this name actually makes utter sense. So it's even though it's a horrendous mouthful, it actually makes every bit of sense. It's a very nice logical name. You can explain every bit of this name. Right. However, because it is a mouthful, it's often abbreviated to HEC for high endothelial cell. Then you miss the associated. Then you put N-acetylglucosamine, and the abbreviation for N-acetylglucosamine is to put GLC, which means glucose, and then N-acetyl. So this is the abbreviation for N-acetylglucosamine. Okay, so even though GLC means glucose, the fact that it's N-acetyl, well, glucose doesn't have an amino group, so the only way that it could have had this N here is if it was glucosamine, so you instantly know it's glucosamine, basically. Okay, so this is just an abbreviation for N-acetylglucosamine. And then you put 6, whoops, not G, 6 for 6 there, and then ST for sulfotransferase. Okay, so it's HEC, gluc, NAC6ST. Okay, but you should read that as HEC, high endothelial cell associated, and then N-acetylglucosamine 6-sulfotransferase. Okay, so what is this enzyme going to do? Well, basically it's going to be expressed in these endothelial cells, and then it is going to work on the glycocalyx, basically. So here is the endothelial cell here, okay, and we have a glycocalyx on the apical membrane of this endothelial cell, which, remember, has a huge number of polysaccharides within it. Now basically what this enzyme is going to do is it's going to add phosphate groups onto the monosaccharides that make up, sorry, not phosphate groups, sulfate groups onto the monosaccharides which make up those polysaccharides on the surface. So basically it's going to sulfonate these polysaccharides. Now why does it want to sulfonate those polysaccharides? Well, basically it's because these naive B cells and naive T cells, they have a molecule on their surface known as L-selectin. Okay, so this is L-selectin. And L-selectin binds to sulfonated polysaccharides. So if you've got endothelial cells which have glycocalyces, which are very heavily sulfonated, which you will end up with if you've got this enzyme activated, then L-selectin can bind to those. Okay, and that's going to promote the recruitment of these naive T and B cells. So naive T, and then I'll put /B lymphocytes. So specifically these naive T and B lymphocytes have this L-selectin on their surface. Okay, so if we sulfonate certain of the monosaccharides within these polysaccharides, then the L-selectin will then have a ligand on the apical surface of the endothelial cells to bind to. Okay, and we'll continue this discussion in the next video.